Today we're going to look at how to make digestion efficient. So far we've looked at the different components of the digestive system, and we've also looked at the enzymes that break down the various substrates such as carbohydrates, proteins and lipids. And we've also looked at enzymes and specifically how enzymes are structured and what factors affect enzyme activity. And in this lesson, we're going to be looking at how the digestive system makes digestion efficient by changing certain conditions. So first of all, we're going to look at the enzyme protease, and we're going to look at how certain conditions are altered to make the activity of protease more efficient. Then we're going to look at the role of bile, and we're going to look at two roles for bile, two, let's say, jobs that bile does, and we're going to explore those two in terms of fat digestion. Here we can see a diagram or a picture of the stomach. Now, when we consider the stomach, we should appreciate that churning occurs where the food is broken down by mixing it around. That's a type of mechanical breakdown of the food. And also we have chemicals that are secreted, such as enzymes, onto the food to help break it down. Now, the digestive system produces many enzymes, and with the aim of speeding up the breakdown of the food that we eat. Now, as we looked at some of the conditions that affect enzyme activity, temperature was one of the factors. And this is why our body is kept at a temperature of 37 degrees. And the body goes to some length to actually maintain that 37 degrees via a process called homeostasis, which we will come on to. Now, our enzymes have an optimum temperature that allow them to work as fast as possible. So that's temperature aside. But the other factor, which we are going to be looking at in more detail right now, is pH. Now, keeping the pH in our digestive system at optimum levels is not as straightforward as keeping the temperature at 37 degrees, because you only have one value which your body tries to work around of 37 degrees at that steady rate. It may fluctuate either side, but it's still a constant value that the body is trying to aim for via homeostasis. But when we talk about pH, we have to now look at it in a slightly different way. This is because we have many enzymes that are involved in digestion, and different enzymes are working at different pH levels. For example, the protease enzyme that is found in the stomach, that is secreted by the stomach, works at its optimal level when it is in an acidic conditions, while the protease that is made by the pancreas works well in alkaline conditions. So we have two types of proteases that are being made, and both have to digest proteins. However, they are made in different parts of the body, different locations, and as such, their pH tolerance is also different. So, before we put this down into words, while you're looking at this image in front of you, I want you to appreciate how the stomach is structured here. And we are going to mention briefly about how the stomach maintains its acidic pH value, and you are going to have to sort of briefly be able to explain that certain cells secrete mucus to prevent the stomach being broken down itself. As you can see here, that the stomach is not just a single layer of protein. It has many folds, it has many substructures as well. And one of the important substructures is where the mucus is secreted from the stomach lining to prevent the hydrochloric acid, which is made in the stomach, to prevent that from eating away at the stomach lining. So we're going to put this all into a few bullet points. I'm going to put this into a few bullet points that I want you to take down as well. So let us take some key points down for the enzyme protease and how its activity is regulated by the stomach. Okay, so the first point is that let's talk about enzymes in general. So the first point is we say that in digestion there are various enzymes. Okay, so various enzymes, not just one enzymes, various enzymes that work best at different pH levels. Okay, so we have various enzymes that work best at different pH levels. So there's not one set pH value that is going to be true for all enzymes in our body. Unlike body temperature, which we can fairly confidently say is 37 degrees and there's minor fluctuations either side, very minor. Now, when we talk about protease in detail, so when we look at protease and analyze the action of protease and its conditions, we can say that, so we'll give this as an example. Okay, example, protease can be produced in two locations. It can be produced in the stomach. So protease is produced in two locations. Okay, A, the stomach, and B, the pancreas. Now, when protease is produced in the stomach, it works best, or its ideal optimal working condition is with a pH which is acidic, which is a low pH value. And this is brought about by the stomach secreting hydrochloric acid. So if we have hydrochloric acid being secreted in the stomach, let's focus our attention now at the stomach area. So what we say is that when we look at the stomach, we say there are a large number of glands lining the stomach wall, and it's approximately 35 to 40 million glands in the lining of our stomach, and these secrete the enzyme protease. And it's a specific type of protease enzyme known as pepsin. So the first thing we say is that, so we're just looking at the stomach. Okay, millions of glands. So we'll just say glands. Glands in the lining of the stomach secrete pepsin, which is a protease enzyme. So glands in the lining of the stomach secrete pepsin. Okay, and this is a protease enzyme, which is going to break down protein. Okay, so glands in the lining of the stomach secrete pepsin. This is the enzyme that is being secreted to digest protein, the protein that we've consumed. Pepsin has an optimal working condition of a low pH value, so works best in acidic pH. So pepsin works best in acidic pH. Now, to accommodate this working condition of pepsin, the stomach secretes a large quantity of hydrochloric acid, a large quantity, and also it's fairly concentrated as well. So to help this, the stomach produces approximately 3 liters of concentrated HCl acid. So the stomach will produce approximately 3 liters of concentrated HCl acid for creating the right conditions for pepsin to work. And you can see here on the diagram, on the lower diagram, why pepsin is important. Why the hydrochloric acid is important for the functioning of pepsin. You do not need to know that pathway in any detail at all for GCSE, but you do need to know that the hydrochloric acid is providing the right conditions for pepsin to work. Also, you need to know that the hydrochloric acid is also very important for killing the majority of the bacteria that is found in our food, so we're not falling ill. Okay, so stomach produces approximately 3 liters of concentrated hydrochloric acid, and this is produced daily so that we can break down the food and make it, and also help with the enzyme activity. So keeping with the stomach and following this on, the next point I want you to note is that the stomach produces a thick layer of mucus. So it produces a thick layer of mucus. Now, this layer of mucus is very important. By definition, pepsin, which is a protease enzyme, is going to break down protein. Our bodies, our tissues, our organs, our muscles, and any connecting parts of our, let's say, ligaments, for example, are all made up of protein. Now, the stomach is a vital organ. It is made up of protein. Therefore, the protease enzyme pepsin will break down protein. And since the stomach is made up of protein, the enzyme is able to break down the stomach, but it doesn't. And in addition to that, the hydrochloric acid, which is fairly concentrated, also, given the opportunity, would break down the stomach. However, this doesn't happen. This is all because of the thick layer of mucus that is secreted by the lining of the stomach. Now you can see here, you've got on the lower diagram, it's got here gastric mucosa. Now these are secreted by so the mucus is secreted by specialized cells in the stomach. Do not confuse this picture with the microvilli that you will find in the small intestine. It looks similar, but do not confuse it with that. But you do need to appreciate that mucus is secreted by the lining of the stomach, okay, and this coats the stomach walls and protects it from being digested by the acid and the enzyme. So the stomach produces a thick layer of mucus, okay. This protects the walls of the stomach, preventing digestion and breakdown of stomach. Okay, this protects the walls of the stomach, preventing digestion and breakdown of stomach by enzyme or HCl acid. So the mucus, the thick layer of mucus, is secreted by the stomach lining to prevent it from being broken down by the concentrated hydrochloric acid, and also to prevent it from being eaten or broken down by the protease enzyme pepsin. So you need to be able to state that the stomach is adapted in one way by producing hydrochloric acid to provide the optimal condition for pepsin, the protease enzyme, to work. But at the same time, it's also producing mucus inside the stomach lining to prevent that acid from breaking down the stomach itself, and also as a protective measure against the pepsin enzyme. Okay, a quick progress check. On Teams, I want you to answer this question back on Teams to me. And can you label it question 1? And I just want you to write down the correct answer. So type the correct answer back to me for this question here. What type of enzyme digests protein? Okay, so you've got an option of amylase, lipase, or protease. So what type of enzyme digests protein? Can you type your answer back to me on Teams? So we're just doing a quick pause here, quick feedback of your understanding. So I want you to type back question 1, and I want you to type back the correct answer. For this question, question number 2, the same process. I want you to type back the correct answer to me on Teams and put it into a private chat so no one else sees the answer. Pepsin is made by the stomach. Name the acid made by the stomach which allows pepsin to work well. So can you name the acid made by the stomach which allows pepsin to work well? Type back your answer on Teams, please. Okay, so now we're going to move on to the pancreas. We've looked at how the stomach is involved in regulating the activity of the protease enzyme. Now we're going to look at how the pancreas is doing this. So can we put the subheading pancreas? Okay. Now we're just going to take a few points down about how the pancreas does this. So when we eat a meal, once it's gone through the stomach and a few hours later after it's been broken down to a certain extent in the stomach, okay, the food leaves the stomach and it moves into the small intestine. So can we just note this down? once food has left the stomach, it enters the small intestine. Now I'm reinforcing the pathway of digestion because you will need to know that for your exam. So once it has left the stomach, the food enters the small intestine. Now, the enzymes that aid in digestion or break down the food in the small intestine are made by the pancreas. So this is the link between the pancreas and the small intestine when we refer it back to the stomach. So what we say is that enzymes that digest food in the small intestine are made in the pancreas. So this is slightly different from the stomach, where protease enzymes were made in the stomach and protein was being digested in the stomach. Here, the pancreas is needed to secrete enzymes that are going to be needed for digestion in the small intestine. The small intestine also secretes some enzymes, but for your exam you will need to understand that the pancreas is vital because it is secreting enzymes that are going to be needed in the small intestine for digestion. So what we say is that enzymes that catalyze digestion enzymes that catalyze digestion in the small intestine enzymes that catalyze digestion in the small intestine are made in the pancreas. So this is important, that you understand that once food leaves the stomach, it enters the small intestine, and pancreatic juices, which will include the enzymes secreted from the pancreas, will also go into the small intestine and help with digestion. Now, as I said, some enzymes are also made in the small intestine. Now, these enzymes that are acting on the food after the stomach all work best in an alkaline environment. So I'll repeat again. These enzymes that are secreted after the food has left the stomach all work best in an alkaline environment. So can you just note that down? So all these enzymes work best in an alkaline environment. Now, this picture makes it a bit more clearer in terms of visualizing the pancreas and its relationship with the small intestine, and specifically how the enzymes are getting into the small intestine. So you can see here you've got the pancreas in yellow, and how there's a pancreatic duct, and you can see how that is leading towards the accessory pancreatic duct. Now, you don't need to know these labels for your exam, but it's good to be aware of how the pancreas is getting the enzymes that are going to be involved in digestion in the small intestine to the small intestine, and you can see that how the connection is made there. Now, what you need to also be aware of here is the role of the gallbladder, and how the gallbladder also is going to now play an important role. The last point that we looked at was how the enzymes that are going to be working in the small intestine all have a working condition where it is alkaline. They need an alkaline environment for them to work well. Now, the gallbladder is going to help in this, because what happens is the acidic liquid that's coming from your stomach, it needs to be an alkaline mixture in the small intestine. As we said, the enzymes that are going to be secreted into the small intestine work well in an alkaline condition. So this alkaline environment is brought about by the liver making a green-yellow alkaline liquid called bile. And bile is stored in your gallbladder until it's needed. So I'll repeat again. The acidic liquid that's coming from the stomach needs to be made alkaline. Now, this is done by the liver making a green-yellow alkaline liquid called bile. And the bile is stored in the gallbladder until it's needed. Now you see how the gallbladder has a connecting duct close to the pancreatic duct. So along with the digestive enzymes that are going to be secreted into the small intestine, you also have bile that is also coming along with it. This is to help aid in the correct condition for these specific enzymes to work now. Now I'm going to show you another picture which puts the bile in its correct, let's say, location with the liver and the pancreas. So here you can see the pancreas, the gallbladder in relation to the liver. So here you can see it's an enlarged image over here. So the image is being enlarged as we look over down towards the bottom left. And notice how when we talk about the bile being created in the liver will create the bile, and when it's needed, it's stored in the gallbladder until it's needed. So as it says, bile drains down the small bile ducts in the liver, and most of it is stored in the gallbladder until it is needed. So as food comes into the small intestine from the stomach, bile is secreted onto the food via the bile duct, and the bile will neutralize the acid that was added to the food in the stomach. So I'll repeat that again. As food comes into the small intestine from the stomach, bile is secreted onto it through the bile duct. Now, the bile neutralizes the acid that was added to the food in the stomach. This provides the alkaline conditions that are needed for the enzymes to work in the small intestine effectively. So let's put a few points down for the role of bile and its relation to pancreatic enzymes. So first point is that bile is created in the liver. So liver makes a green yellow alkaline solution. Okay. And this green-yellow alkaline solution is called bile. Okay, so there are a few points here. One, bile is alkaline, it's made in the liver, and the color of it is greenish yellow. Next point, so we'll call this A. Let's call this A. Okay, so the next point is that bile drains down. So once it's made in the liver, bile drains down small bile ducts, okay, and stored in the gallbladder. So can we say that bile is stored in the gallbladder until it is required. So it's made in the liver and then it is stored in the gallbladder until it's needed. Now, when it is needed, it's secreted onto the food via bile ducts. Okay, so when needed, bile is secreted onto the acidic mixture via the bile duct. And what is the result? The result is that that mixture now becomes alkaline. Okay, so a quick break. So we're just going to have some quick questioning here. And again, as before, what I want you to do is look at these two exam questions and type your answer back to me on Teams in a separate chat box so no one else sees your answer. So question 3, you're going to have to identify correctly the various components of the digestive system. And question 15, again, it should be straightforward. This goes back to the initial topic of cellular organization. So question 3 and question 15, can you pause the video here? Can you send the correct answer back to me on Teams? Now the second function of bile is shown here. Now proteins and carbohydrates are broken down via mechanical and chemical means. And by the churning of the stomach and the action of various enzymes, we can very easily break down carbohydrates and proteins. However, lipids and fats, they do not easily mix with water because they are hydrophobic by nature. And a lot of our digestive system secretes a lot of water mixtures. Now, this is not going to affect hydrophobic molecules such as fats. Therefore, we need to get the large fat molecules into smaller ones. Now, yes, the lipase enzymes do break down the fat molecules. However, when we consume fats, they are too large for the enzymes to work effectively. So what happens in essence is that we have bile being used. So bile is used to emulsify, to break down. Not break down, it emulsifies the large lipid molecules. Now, so it is important that you understand that lipids are emulsified, fats are emulsified by bile. Bile does not break down large fat molecules, it emulsifies it. Now, the breakdown of, let's say, lipids into its smaller constituent pieces happens via the enzymes. But for the enzyme to work, we need a bigger surface area for the enzymes to work on. So what we say is that the bile will emulsify the fats in our food. This means that the bile will physically break up large drops of fat into smaller droplets. So this is simply just separating it out from a large globule to smaller globules. This provides a bigger surface area for the lipase enzyme to work, and the enzyme itself will start breaking down the fat molecules into its fatty acids or glycerol, as we looked at before, as we looked at how to make up fats. The enzyme will break it down into its constituent parts. So you can see here how large lipid droplets or globular lipids are emulsified into smaller droplets. And there's more information here than you need, but you do need to appreciate that fat droplets or large lipid droplets are emulsified into smaller droplets via the action of bile. So bile is emulsifying large lipid droplets into smaller lipid droplets. This allows for the pancreatic enzyme lipase here to start breaking down our smaller droplets of lipids into their constituent parts that you can see here and that are going to be absorbed through the mucosal cells. So this is important in terms of understanding that the second aspect of bile is to emulsify large lipid droplets into smaller droplets, giving a greater surface area for the lipase enzyme to work on. And the enzyme will break down the lipid molecule or the fat molecule into its constituent parts that you can see here and allow for easier absorption. Okay, so let us just put this into a few words. So can you just put down bile is not an enzyme. Okay, so this is the first point. Bile is not an enzyme. Okay, it does not break down fat molecules. Yeah, I think it does not break down fat molecules. Okay. Bile will emulsify. Bile will emulsify the fat into smaller droplets. Of fat. Okay, so bile will emulsify the fat into smaller droplets of fat. Okay, this increases the surface area for lipase enzyme to digest or break down the fat molecules. So therefore, increasing the surface area for the action of lipase. Okay, so bile essentially creates a larger surface area for the enzyme lipase to work.